Well, hello again, students. Today we want to look at the cell membrane and transport systems. Now, for many of you, you're listening to this and you're probably somewhere along your university career. In most cases, I suspect you're at the beginning of your university career and in your first year. And chances are this is a topic that you would have studied extensively in high school. So much of what we're going to cover today, I suspect, is going to be revision for you. What I'm going to do then is probably move fairly quickly, but highlight certain things that might be new and focus on things that have more clinical relevance. As always, when I have my lectures, particularly for my students at the University of the West Indies, I like to put up a list of learning objectives. And these can serve as something you can use for revision. If you can answer these as questions at the end of the lecture and at the end of your period of study, then you can safely say you've done well and you're ready, you know your material. So let's just talk a little bit about the cell membrane. The cell membrane is the structure that forms the wall of the cell or the outer boundaries of a cell. And all cells in the human body and the animal body have a cell membrane. It's also known as a plasma membrane. And it's an essential part of life because it separates the cell and the internal contents of the cell from the outside environment. And previously, we would have spoken about homeostasis. And the cell membrane is an integral part of the systems that the body has developed to maintain homeostasis. And what it allows is it allows the contents of the inside of the cell to be different from the contents of the outside of the cell. And that's why we talk about the intracellular fluid and the extracellular fluid. Two totally different fluids that allow different functions to be performed inside of the cell versus outside. When you get within the cell, there are organelles, and these organelles also have membranes. And we can talk about the issue of compartmentalization. And here the membranes serve the same function. It allows the environment inside of the mitochondria to be kind of different, slightly different from the intracellular fluid. A good analogy and a good way to think about this is to think about a house with its walls. The outer walls of the house divide the external environment from the inner environment of the house. And then within the house, there are individual rooms, which are like the organelles or the compartments of the cell. And the environment of your room may be very, very clean and very, very orderly and very different from the rest of the environment of the house. Though maybe as young teenagers, as young students, it could be the opposite. Now, I'm sure many of you are familiar with this structure. The cell membrane is a phospholipid bilayer. It primarily consists of phospholipids. These are molecules that have a phosphate head and a two-tailed lipid tail. When they're placed in solution, they aggregate into a bilayer, which gives the underlying architecture for our cell membrane. The lipids form the vast majority of the surface area of the membrane. But actually, if you were to look at it by weight, embedded in the lipids are proteins. And the proteins actually form a greater proportion by mass of the cell membrane, maybe approximately 55%. So the two major components of our cell membrane are the phospholipids and the proteins. And this diagram here that you see on the right just gives us a little bit more detail of the structure of the phospholipid. The head, it is able to interact with water. And so this is the part that you find on the outer surface of the membrane and the inner surface of the membrane. The tail is hydrophobic. It doesn't like water. And that's what you find in the center of the membrane. And like I said, these can spontaneously aggregate to form this bilayer structure, which is the core of the cell membrane. And this is just shown to you here in this diagram. You see the structure in figure A. Then you can see it forming a monolayer. But if you shake it up, you get a bilayer structure, which is classical for the cell membrane. Now, inside of that bilayer structure, we just said there are proteins embedded. And we classify these proteins in two ways. We talk about integral proteins and peripheral proteins. Integral proteins are designated because they're usually attached to the cell membrane and they can't be removed. The vast majority of these are what we call transmembrane proteins. They span the entire length of the membrane. And you can see an example in this diagram here. This would be a transmembrane protein, which also forms a channel and allows substances to move in and out of the membrane. But some proteins are attached to the inner surface or the outer surface. These proteins tend to be very mobile, and and we call those peripheral proteins. And so those are the two classes of proteins, those that are fixed and are usually transmembrane and integral, and those that are just found on the surface and have the ability to move and aren't quite as fixed. We call those peripheral proteins. Now, collectively, this structure forms something called a fluid mosaic model. What you see here is an experiment in which a number of the proteins were tagged with a fluorescent dye. Some were tagged with a pink dye, and some were tagged with a lime green dye. Once they were tagged, a period of time was left. And you can see that over time, these proteins actually moved around until they were spread out throughout the cell membrane. So what does this tell us? Well, it tells us that the proteins are mobile or fluid. And it also tells us that at the end of the story, we have a very mixed structure. It looks like a mosaic. And that gives us the name, the fluid mosaic model. And as I always encourage my students, I like you to understand your work. And so you don't just learn what a fluid mosaic model is, you understand why it has a particular naming. So if you ever forget, you can just think about those words and you can make sense of it. These are just a list of some of the major phospholipids that are found in the cell membrane. I'll just let you look at those and learn those. You can read about these in your book. I just want to point you towards one important other structure of the membrane, and that's cholesterol. Now, cholesterol is often found in high concentrations in the cell membrane, maybe even in a 1-to-1 ratio. And cholesterol acts as a stabilizer for the membrane. What it does is it stops the molecules becoming too deformable and sliding by one another too much. If that were to happen, the wall would not have any strength or functionality. And then the cell would actually need a cell wall like it happens in bacterium. So what you find is this cholesterol acts as a stabilizer and affects the fluidity of the membrane. At high temperatures, it stops the membrane becoming too fluid. Whereas low temperatures, it stops the membrane crystallizing and basically becoming deformed and non-functional. And so cholesterol is a very, very important component of the cell membrane. Well, what are the functions of the cell membrane? The more common or significant functions are, as I mentioned already, it provides physical isolation between the inside and the outside environment. It regulates the exchange of substances between the inner and the outer environments. It acts to detect changes in the environments, both on the inside and the outside. And it can be used to form structural support as cells come together to form tissues and organs. And these are the major functions of the cell membrane. And often these are achieved by the proteins that are embedded within the cell membrane. And I'm not going to go through this in detail, but you can see the functions there again are transportation, movement of substances in and out. Sometimes these proteins can function as enzymes to trigger reactions that are important in the cell. They can function as receptors or detect changes outside of the cell. we see that these can also function for structural support and cell-to-cell recognition and attachment to the cytoskeleton and the extracellular matrix. So all of these things are done by the proteins that I just mentioned. So that's a quick summary of the cell membrane. And I suspect you might have done that in a little bit more detail in your high school career, or some of you may have even studied a first degree. Now we want to talk about, well, what are the processes that the body uses to move substances across cell membranes? Let's just remind ourselves that the cell is permeable to very small non-polar molecules like oxygen. It's permeable to small polar molecules like carbon dioxide and to some extent water a little bit. And it's also permeable to lipid-soluble molecules like steroids. However, large molecules that are polar charged like glucose and highly charged inorganic ions like sodium and potassium, the membrane is not permeable to these substances. However, these substances are very important for the cell. For example, glucose, we know, is the main substrate for energy in the body. And that process of oxidizing glucose takes place within the cell. So the body has to develop a system that allows glucose to get into the cell. And that's what we want to talk about. Other than simple diffusion of these smaller molecules, how does the body move substances across the cell membrane? Well, we're going to look at four systems or four processes by which this occurs: diffusion, osmosis, active transport, and vesicular transport. So diffusion is something you know well. All molecules are in a state of constant random motion, and we call that Brownian motion. Those of you who have studied chemistry should be familiar with that. And because of that process, because molecules are in a constant random motion, whenever you have an area of high concentration of these molecules, they are going to, just because of their random movement, move to an area of low concentration. So you can put something that's very pungent and has a very strong smell in the corner of a room. And over time, that substance will diffuse throughout the room, and eventually you'll smell the scent at the opposite end of the room. This is going to occur until there's an even concentration throughout the vessel. At this point, net movement ceases, even though the particles are still in constant random motion, meaning the movement from side to side, the overall net movement has stopped, but the molecules are exchanging themselves back and forth. Now, one of the things we're interested in biology is the rate of diffusion. For example, in the lungs, how quickly can the oxygen get out of the lungs through the membranes of the alveoli, through the membranes of the capillaries, and into the blood to be circulated throughout the body? In the same vein, how quickly can carbon dioxide move in the other direction? That exchange of gases at the alveoli and the capillary membrane inside of the lungs takes place as a result of diffusion. And we want to know what are the factors that affect the rate of diffusion, because that might have implications in pathological conditions. And there are 5 issues or 5 factors that you need to think about when you're considering the rate of diffusion. The first is the concentration gradient. The larger the concentration gradient, the greater the rate. The second is the permeability of the membrane. The greater the permeability, the greater the rate. And the permeability is dependent upon the physical characteristics of the membrane and the physical characteristics of the substance. And so each substance will have a different permeability coefficient with regards to a particular membrane. The third is temperature. You and I know that temperature affects Brownian motion, and therefore an increase in temperature will cause an increase in the motion of particles and therefore lead to an increase in diffusion. The fourth is surface area. The larger the area of the membrane available to the substance, the more quickly the substance can diffuse through the membrane, and therefore that increases the rate. And finally, the thickness. And here we have some inverse proportionality. In this case, the greater the thickness of the membrane, the longer it's going to take for the substance to diffuse. And that's how it's going to affect rate of diffusion. Now, you know, I'm focusing a lot here on diffusion through membranes. Diffusion doesn't have to just take place through a membrane, but in terms of the human and animal bodies, that's what we tend to focus on. Now, this has been put together in a formula called Fick's law. And you can see there that the rate is equal to the negative of the diffusion coefficient multiplied by the area of the membrane, multiplied by the concentration difference on the inside minus the outside, divided by the thickness of the membrane. And that's an equation you should know. Now, we can simplify this equation for biological membranes because biological membranes all have the same thickness approximately. And in biology, at human body, most of the body is at the same temperature. So we can simplify that equation by bundling the thickness of the membrane and bundling the temperature and bundling the diffusion coefficient into something which we call the permeability coefficient or the permeability constant. Once we have that, the formula simplifies into the permeability constant multiplied by the area by the concentration gradient. And notice in this case, it's the outside minus the inside. So we're able to remove the negative sign. And you should be able to do simple calculations using these equations. And you should also be able to explain why these factors affect the rate of diffusion and draw graphs to explain these things. Now, sometimes you have a large molecule in a high concentration outside the cell membrane, and it wants to diffuse down the membrane, but it can't. It can't pass through the membrane by a simple diffusion. In this case, we can use a carrier protein to shuttle the substance across the membrane. When this happens, the protein undergoes a conformational change. It binds the substance on one side, changes its conformation, and transports the substance to the other side. And we call this process facilitated diffusion. Now, please note that in this case, the movement is still from the area of high concentration to an area of low concentration. And so because of that, no energy is required. But what is required is a transporter or a protein. And this is often called protein-mediated transport or facilitated diffusion. And this diagram here just shows you what I was trying to capture. Shows you very nicely an example of facilitated diffusion or protein-mediated diffusion. This is our carrier protein, and it receives the substance on one side and then undergoes a conformational change and is able to release the substance into the inside of the cell. Now, because of the fact that you require a transport protein to manage facilitated diffusion, the rate of diffusion obeys different laws. The kinetics is a little bit different. Now, on this graph, we're looking at diffusion of glucose into the cell. And generally, glucose cannot cross the cell membrane. So because of that, even though the concentration gradient is increasing, there's only a very slight increase in the amount of glucose that moves across the cell membrane. However, when you have a transporter present and the concentration increases, notice we have a much greater increase in the rate of diffusion. Actually, this is the role of insulin in the body. When you have a meal, your body releases insulin. And what insulin does is it causes the glucose transporters to be inserted into the cell membrane. And as a result, you're now able to achieve facilitated diffusion. The other thing to note about this graph is it reaches a maximum. It plateaus. It doesn't keep increasing as concentration gradient increases. And that's because a point is reached when the carrier proteins are saturated. And at that point, no matter what you do with the concentration gradient, you still cannot increase the rate of diffusion. And so this tells us that when we look at protein-mediated transport, there are certain characteristics. It's specific, meaning the transporter will only interact with one molecule. Competition can occur. A molecule can compete with glucose and block the carrier protein. And finally, it demonstrates saturation kinetics with a transport maximum when the carrier sites have become saturated, as I just explained to you. Okay, so we just looked at diffusion, and we looked at simple diffusion and facilitated diffusion. Let's talk a little bit about osmosis. Now, osmosis is the movement of water. It's only the movement of water. And it's the movement of water across a semi-permeable membrane. And it's a movement of water from a region of high solute concentration, from a region of low solute concentration to a region of high solute concentration. So I want you to keep those three things in mind anytime you say the word osmosis. One, osmosis only refers to water. Sodium does not move by osmosis. Glucose does not move by osmosis. Osmosis refers to the movement of water across a semi-permeable membrane. And osmosis is a movement from a region of low solute concentration to high solute concentration. And that's demonstrated in this experiment that should be very familiar to you. Here we have two solutions, one with low solute concentration, one with high solute concentration. This membrane is impermeable, so the solutes cannot move. Therefore, in order to create balance and in order to achieve the same concentration on both sides, the water moves. And you get this very unusual situation where one side of the tube, the height of the water, is different to the other. And that's due to the osmotic pressures generated by those impermeable solutes. That movement of water is called osmosis. Now, just a couple of things I want you to note because you'll see many terms in your books and I want you to be clear about the terms. Now, osmolarity is the number of particles per liter of solution. So you'll see in your books the word osmolarity. It's a number of particles per liter of solutions, and we talk about millimoles per liter. And that's what we tend to use more often in medicine. But you'll also see in your books the word osmolality. And this is the number of solute particles per kilogram of water or per kilogram of solvent. And therefore, we can talk about millimoles per kilogram. Now, in terms of measurement, the thing that we tend to measure is osmolality. But like I said, the thing that we often refer to in medicine is osmolarity, because we like to deal with volumes in liters, not kilograms. For most biological systems, they're very closely related, and you can see the formula there. The osmolarity is 0.995% of the osmolality. And so that's why we tend to use them interchangeably. But I do want you to recognize the difference. The osmotic pressure of a solution is a measure of its tendency to take in water via osmosis. And often you'll hear us using the term oncotic pressure in biology and in human physiology and animal physiology. And when we're using that, we tend to refer to the osmotic pressure generated by large insoluble proteins like albumin that's found in the plasma. So oncotic pressure is a specific example of osmotic pressure. Now, just building on this concept a little bit, we can talk about milliosmomoles and millimoles, and there is a slight difference. The milliosmomoles is the index of the concentration of particles per liter of solution. So if you have 150 millimoles of sodium chloride, that splits up. You know, when you dissolve sodium chloride in water, the result is you get an ionized solution of sodium and chloride ions. So 150 millimoles of sodium chloride dissociates to give you 300 millimoles of, well, 150 millimoles of sodium and 150 millimoles of chloride. And that actually is 300 milliosmomoles. A substance that doesn't dissociate, like glucose, 300 millimoles of glucose will yield 300 milliosmomoles of osmotic pressure. So just reminding you of these things, And when you do calculations, you'll come to appreciate these things a little bit more. Now let's talk about the word tonicity. Tonicity. Tonicity is the ability of a solution to gain, to cause a cell to gain or lose water. It's determined by the number of impermeable solute particles that are present in the solution. And to explain this, we'll just use a classic example of a red cell placed in a solution. Now, generally, we think about the inner solution of cells as being about 300 milliosmomoles. So if you place a red cell in an isotonic solution, that means the solution is also about 300 milliosmomoles. And the result is the amount of water entering and leaving the cell will be the same. And so the volume of the cell will not change. If we place the cell in a hypertonic solution, that's a solution that has a greater osmolarity, say 400 milliosmomoles. The result is water is now going to leave the cell. And that's why we call it hypertonic, because it's going to cause water to leave the cell. And the cell is going to become shrunken and shriveled. If we place the cell in a solution that's lower, say 100 milliosmomoles, well, in this case now, water is going to enter the cell and it's going to cause the cell to swell. And depending on the concentrations, it might even cause the cells to burst. And so when thinking about the term tonicity, we're thinking about two things. We're thinking about what effect does that solution have on a cell? Does it cause a cell to gain or lose water? And we recognize that it's determined by the number of impermeable solute particles. And you're going to see why that is so important in a couple of minutes. Now, you might think tonicity and osmolarity are the same, but there's a very subtle difference that I want you to recognize. The osmolarity is dependent on the total solute concentration, whereas the tonicity is dependent on only the impermeable solutes. So I want you to lay hold of that. A solution can have solutes that can cross the cell membrane and solutes that cannot cross the cell membrane. Tonicity is determined by solutes that can not cross the cell membrane. Osmolarity is determined by adding both together. So this is just a little summary table telling you the difference between tonicity and osmolarity. And I want you to take your time and think about these things and consider them. You can pause the video here and look at these things and read it. And we're now going to look at an example where two fluids can have equal osmolarity but a different tonicity. And I'm going to show you an example where a fluid could be hyperosmotic but isotonic as well. So let's look at that here. Imagine you have a solution that contains 300 milliosmomoles of sodium chloride, right? Well, sodium chloride cannot cross the cell membrane, so it's impermeable. But that solution also has 100 milliosmoles of urea. The total osmolarity of that solution is 300 plus 100, which is 400. Okay. Now, compared to the cell, 400 is greater than the 300 that you find inside the cell. So we can say that that solution is hyperosmotic. However, only 100 milliosmomoles of urea is permeable. The other 300 milliosmomoles of sodium chloride is non-permeable. So when you place the cell into that solution, what's going to happen is the urea is going to diffuse and distribute itself between the intracellular and the extracellular fluid. And it's going to reach a balance point where the size of the cell will not change. Initially, what will happen is when you place the cell in the solution, because the solution is more concentrated, it's hyperosmotic, water is going to leave the cell and it's going to shrink a little bit. But as the urea equilibrates, the water is going to move back and the cell is going to return to its same size. So that solution that we're looking at is isotonic, but hyperosmotic. Initially, there'll be a transient shift in fluid, but when everything equilibrates, there'll be no change. And that's simply because urea is able to cross the cell membrane. It affects osmolarity, but it doesn't affect tonicity. I want you to pause. I want you to think about this. I want you to look at other videos, and I want you to consider this and lay hold of this concept. Important concept, and one that I hope you can understand. Okay, let's just conclude by looking at a couple of other means of transportation, and these are things that you know well. Let's look at active transport. Well, active transport is the movement of a substance against its concentration gradient. It's a movement from a region of low concentration to a region of high concentration. And because you're utilizing, because you're going against the concentration gradient, it's always going to involve the utilization of energy. And that energy in biological systems comes from ATP. So in this diagram here, we see we want to move this substance in green against its concentration gradient. It requires a carrier protein, and that carrier protein is going to have to utilize energy to achieve that movement. Now, active transport is divided into two, primary and secondary. Primary active transport is when the energy for the movement comes directly from the hydrolysis of ATP. And the classic example of that is the sodium-potassium ATPase pump. Now, this pump pumps 3 sodium molecules out of the cell into the extracellular fluid against its concentration gradient. And it brings 2 potassium in the opposite direction against the concentration gradient. And it's achieved by our pump that not only is it a pump, but it also hydrolyzes ATP. It's an ATPase. So this complex protein is an enzyme that hydrolyzes ATP to release energy. And it's also able to shuttle 2 ions against their concentration gradient. So here you see the energy for this system is coming directly from the hydrolysis of ATP. And we call that primary active transport. Now, ATP can sometimes be used to set up potentials and potential energy. So when ATP is used to set up a concentration difference, just like we saw with the sodium-potassium ATPase pump, that concentration difference is potential energy. The sodium being in high concentration on the outside of the cell versus the inside is potential energy. And when sodium moves down its gradient, its concentration gradient, that energy is released. And if you have the right transporter, it can harness that energy to move another substance against its concentration gradient. And that's what you're seeing here for the movement of glucose. As sodium moves down its concentration gradient, glucose is used to move in the opposite direction against its concentration gradient. And here we call this secondary active transport, because the potential energy that was set up by the hydrolysis of ATP is now being used for active transport. It's not directly being used, it's coming from an indirect way. And I want you to go and look up some other examples of secondary active transport. And finally, vesicular or bulk transport. Well, in this case, you're using the formation of vesicles, very small little buds, where the substance doesn't actually cross the membrane itself, but a new membrane forms around the substance. I think many of us know of exocytosis, where here you see a vesicle coming and it has contents, and due to receptors, it interacts with the membrane, it causes the membrane to lyse and release its contents. And so exocytosis means the bulk movement of substances out of the cell. And in this case, a very good example that you can study or think about is neurotransmitters. Well, the opposite of that is endocytosis. Here you have the movement inside of the cell. And that could be a solid. And if it's a solid, we call it phagocytosis, cell eating. And I'm sure you remember the classic example from high school when you study the amoeba eating the paramecium by surrounding it and forming a vesicle. You can also have pinocytosis or cell drinking, where you encapsulate fluid and form a vesicle. And very often these processes are triggered by receptors that interact with the substance on the outside. And in this case, we'd call that receptor-mediated endocytosis. So there you saw the examples of bulk transport or vesicular transport. Very simple examples, very simple systems that the body uses to move larger substances in and out of the cell. Finally, this is just an example of all the different types of molecules and how, of all the different types of transport and some molecules that use these different types. I'm not going to go through this. You can simply pause the video and just look at them. And of course, all of these things can be found in your textbook. And so that brings us to the end of our study of cell membrane and transport systems. Like I said at the beginning, this is something you've probably studied before, but now you can begin to think about these things from a clinical perspective. So for example, understanding factors that affect the rate of diffusion might be important if you have a lung disease or kidney disease that changes the amount of diffusion of oxygen or the amount of diffusion of glucose in the kidneys. Active transport can become very important in the nervous system. And if you have something that blocks the active transport of neurotransmitters, that can affect how the brain works and how nerve works. So understanding these systems allows us to understand pathology and also allows us to develop drugs and different systems that can help us to improve function and to eventually improve life for people who are ill. So let me end there and say goodbye.